Hopefully you remember the last lab, which was an introduction to gel electrophoresis. In this lab, we are going to be continuing our study of electrophoresis, but we're going to be determining whether or not a family has sickle cell anemia. As you read in the introduction, sickle cell is a genetic disease, which means that the genes of those who have it will be different, and thus the disease will cause alterations in the DNA. When you perform gel electrophoresis, different DNA shows up differently. If we have a DNA sample from someone we know has sickle cell, we can determine if another patient also has sickle cell. Specifically, running DNA through a gel creates a banding pattern. If our patient has the same banding pattern as a control who we know has sickle cell, then that means our patient also has sickle cell. As I mentioned, in this lab we are going to be studying a family, and we're going to be determining whether or not a mother, child, and father all have sickle cell. Like the last lab, I'm going to be loading samples into the top of the gel where the wells are. You're going to want to record the samples I'm loading on your handout so you can keep track of the results when we review them. So for the first two lanes, those are typically reserved as practice for you all. While I've loaded many gels, practice never hurts, so I'll be using these for practice as well. The next well, which is lane 3, will contain a sample from the individual that we know has sickle cell. In lane 4, we'll have a sample from a sickle cell carrier. Remember, a carrier does not express the disease, but they can pass it on to their children. In the next well, which was lane 5, we'll have a sample from an individual we know to be healthy. In this case, that means they neither carry nor express sickle cell anemia. And then finally, in the last 3 wells, which are lanes 6, 7, and 8, we'll have samples from the mother, child, and father, respectively. Remember, we don't know yet whether or not these individuals have or carry sickle cell anemia. So running their DNA through the gel will allow us to determine this. After the samples are loaded into the wells, I'll place the gel into the gel box where the electrical current will separate the various fragments. Once that's completed, I will actually need to apply a dye or a stain so we can see the banding pattern more easily. Once those 2 steps are completed, we'll go over the results. So let's get started.
I'll narrate the process of loading the first well, although this should be very familiar to you from our last lab. So first I'm going to put a tip on the pipette. Remember, you can't use a pipette without a tip. Now I'm going to locate the sample. Remember, for the first 2 wells, it's just a practice dye, not a DNA sample. I'm also going to adjust the volume the pipette will be extracting by twisting the button on the top. To draw solution into the pipette, first I'm going to push the button down, then place the tip of the pipette into the sample, and finally release the button. Once the pipette is positioned into the well at the top of the gel, I will push down the button, which releases the sample. Before I lift my thumb, I will remove the pipette from the well. Otherwise, I would draw the sample right back into the pipette. After I finish with this first sample, I'm going to eject the tip. It's essential for this demonstration to change tips between each sample, otherwise contamination may occur and the DNA samples could get mixed together. I'm going to repeat this process 7 more times to fill the other 7 wells. Remember, the second well will also contain practice dye. Well 3 will contain DNA from a sickle cell patient to serve as control. Well 4 will contain DNA from a patient carrying, but not expressing, sickle cell. Well 5 will contain DNA from a healthy individual that does not have sickle cell. And wells 6, 7, and 8 will contain DNA from the mother, child, and father, respectively.
Now that the gel is loaded up with all the samples, I've placed it in the gel box where it will be exposed to an electrical current. If you look carefully, there are some bubbles rising at the far end of the gel box. You should also be able to see the samples up at the top near the black band. Again, our current is being split into positive and negative charges. The negative charge is coming through the black wire at the top of the gel where our samples are, and the positive charge is coming to the far end of the gel through our red wire. The reason why the charges are split in this way is because the DNA samples are negatively charged and we want them to move down the gel. So giving the far end a positive charge draws the samples to that side. I'm going to let the gel sit in the box and be exposed to the current for 35 minutes. By the end of this time period, all of the samples should be separated out into their respective DNA fragments. Then I'm going to stain the gel and we'll take a look at the results.
Okay, so we've finished running the gel and now we need to stain it. So if you can see, our staining indicator has moved about halfway, maybe a little bit more down the gel, but we can't actually see the bands right now. So I'm going to add this stain, and the whole process is going to take about 25 minutes, which I'll skip past for you guys. And then once we do that, we should be able to see the bands a little bit better. Okay, so the samples have been allowed to run through the gel for 35 minutes and they've been stained. So we're ready to look at and discuss the results. I know you probably can't see the gel that I have here, so I'm going to put some pictures up next to me so you can look at what I'm talking about while we go through the results. So remember, the first two lanes were just for practicing loading, so you can't really see any bands here, and they're not really relevant to our sickle cell investigation. So moving on to lane 3, we had our patient that is known to have sickle cell, and their DNA resulted in 1 band. In the next well, lane 4, was our individual that was known to carry but not express sickle cell. Their DNA resulted in 3 bands. In lane 5 was our healthy individual that neither carries nor expresses sickle cell, and their DNA resulted in 2 bands. So now we just need to look at mother, child, and father. We will know what their sickle cell status is by comparing their unknown banding patterns to our known banding patterns and seeing which ones match. So I'm going to let you figure that out on your own and record it on your handout. If you need help, your teacher would be more than happy to provide further guidance. Don't forget, you also need to draw the banding patterns and record each family member's genotype. If you need additional help with this, go back to the introduction slides. So I hope you enjoyed this lab and learned more about DNA, genetic diseases, and electrophoresis.